Well, thank you very much for that kind introduction, Julie, and thank you all for coming this evening. Thanks for the Cabrillo Aquarium for having this series. It's been a while since I've been able to come, so I'm really excited to be here this evening and share with you some of the work that primarily my students have been doing. As we faculty, we get a lot of the credit, but of course it's really the students that are the ones that are out there in the ocean doing the research, and so I'll highlight some of those students. Tonight's talk is actually a culmination or the sort of an update on 3 different master's students as well as some undergraduates that have worked with me, and I'll highlight that. But before we get to kind of what we're doing today, I thought I would give some context into microbial biofilms and microbial mats. And to do that, we can really go back to the beginning of our time here on Earth, and really not our time, but the time of microbes on Earth. So the geologists tell us that the Earth likely formed around 4.6, that's G as in giga annum, which means billion years. Okay, so the Earth is very old. I think we can all attest to that. And this diagram on the right-hand side, this diagram on the right-hand side sort of shows if we were to take a 24-hour or 12-hour clock and break it down into the history of life on Earth and the history of the Earth itself, our human existence on this planet really is very brief and only comes in at the very end. But the microorganisms likely evolved on this clock around 9 minutes or maybe something like 13 minutes. And we'll talk a little bit about some of the evidence for that. But the main point I want to get at here is microbes have been on this planet and in the world's oceans for a very, very long time. And after we're all gone from this planet, hopefully not very soon, but in millions of years from now, there will be microbes on this planet still doing their thing. And that's really the thing that has drawn me over my career to studying the microbes, especially with trying to understand using modern versions of the microbial communities to better understand what life may have been like way back when. But of course, we didn't come on the stage here until the very last, almost second on this clock. So it just kind of puts that in context. And really, we use a number of terms when we talk about the organisms on our planet. So some of you may be aware that we use the term prokaryotes versus eukaryotes, right? So we are eukaryotes. We have nuclei and organelles and things inside our cells, whereas the bacteria and then the other major group on the tree of life, the archaea, have likely been around a very long time, and they're primarily unicellular organisms. And so for much of the 3+ billion years of life on our planet, it's been a microbial planet. And only more recently have we ended up with what we see today in terms of plants and all the wonderful animals, including ourselves, running around the world. And so this just gives you kind of a general overview and some view. Obviously, when you look back in geological time this far, the Earth has changed a lot, and I think everyone in this room is probably well aware of terms like Pangea and the movements of the continents around on our planet. And you know, that on a microbial time scale has impacted them very much. But the microbes have adapted to pretty much any environment on this Earth since they first arose those billions of years ago. And in fact, when life first evolved many years ago, things were a lot rougher, right? They were very harsh, the early Earth's conditions, at least from a human perspective. Now remember, we shouldn't put too much on the microbes that way, because there are many microorganisms that prefer to be in extreme temperatures or extreme salinities or extreme pressures that would crush us in a matter of moments. But they evolved in these conditions starting back when the Earth first cooled enough to probably have the conditions suitable to life. And we don't know exactly when that happened. We don't know exactly what that looked like. But we do know that the chemists and the geologists tell us that the conditions back then were very different. We didn't have an ozone layer, and I hope everyone in the room is aware of the importance of the ozone layer to drop the UV radiation that allows us to survive, right? So there was very little oxygen, and ozone is O3, and is derived from O2, or oxygen. So the amount of solar radiation impacting the surface of the Earth was profound, and that may have resulted in the oceans being the refuge or the so-called cradle of life. We don't know where life first evolved on our planet, but it's quite possible that it was in perhaps near the surface or maybe way down deep, and tonight's lecture will kind of explore some of these topics a little bit. It was also much hotter at that point, and the continents were far fewer in size and number at that point. So what is the evidence for how life first evolved? It's old, so it's not perfect, let's put it that way. But we have these fossils from certain places on our planet, and these are particularly places like Greenland and Australia, which have not been subject to subduction and plate tectonics, where the land masses have been taken back under the Earth, destroying the fossil record. So in places like Greenland and Australia, there are some very, very old, multi-billion-year-old rocks, and we can go to those locations, or the paleogeologists can go to those locations, and this is what they find. They find things that form sort of wavy, laminated structures here, like these stromatolites, or they find things that look like these kind of filamentous structures that are thought to be microfossils, potentially the remnants of early life, and these are over 3 billion years old. So we think that, we don't know what that first sort of primordial ooze was, but we think that very rapidly, over 3.5 billion years ago, we ended up with these communities forming. And they likely formed these stromatolite structures, which we now equate with modern microbial mat structures, okay? And so these are communities. It's not just a single organism out there on its own. It's living with other organisms. And that makes a lot of sense if you think about it, because we as human beings certainly do better collectively, but all of life really relies on other organisms to help feed and sort of house and create the conditions favorable for suitable life. So this is some of the primary evidence. There's other lines of evidence I won't go into this evening, but it gives us a sense of what life may have been like back then. And I'll point out that, many of us that care about marine biology find coral reefs to be fascinating structures, right? Well, stromatolites can really be thought of as the first type of coral reef. As you know, coral reefs are photosynthetic structures. They have calcium carbonate structures that give them that hard structure that allows these amazing communities to survive. Those didn't exist 3 billion years ago, but these stromatolites did. And they're also made of calcium carbonate, the same structures that are found in modern reefs. And so we think that these were likely very plentiful in shallow seas and portions of the ocean at that point. Now, I mentioned that microbes, just like us, like to live together, and so we use the term consortia or consortium to refer to that. And you can see many microorganisms that live together, many examples of this. And it enables them, as I mentioned, to exchange things like nutrients or vitamins, things like this. And of course, we are well aware, I hope you're all aware, that you have microorganisms that live inside your gut and really all over your body. And many of them are very beneficial to us, and we wouldn't be able to digest our food without them, et cetera. Well, most of the microorganisms on our planet don't really care about human beings, I hate to say, but they do interact with each other and with other plants, animals, algae, and other microorganisms on the planet. And the sort of ultimate consortium really are these microbial biofilms, or microbial mats, as we call them, that are often filled with an amazing diversity of microorganisms. My postdoctoral mentor used to refer to microbial mats as a rainforest in a teacup, because you can fit the same amount of diversity of bacteria and other microorganisms in a very small amount that you would find in something as amazingly diverse as the Amazon rainforest. So I mentioned that coral reefs are sort of equatable with the stromatolites, but of course we don't see stromatolites in that many places on Earth today. And there's a number of reasons for that. Things like to eat microbial mats because they're full of carbon and nitrogen and other essential nutrients. So they're really mainly found in what we'd refer to as extreme habitats now, right? So things like coral reefs have replaced them in the world's oceans, and so you have to go to really exotic places like the Bahamas or northern Australia to find some of these stromatolitic structures in the ocean. And then if we go to other locations such as this saline intertidal flat down in the Baja California, or this mushroom hot spring in Yellowstone National Park, where I did a lot of my doctoral work, you can see these microbial biofilms living there, where because of the extreme conditions, they don't get eaten by grazing organisms like snails and invertebrates and other things that would eat them today. So we go to these extreme habitats and study these microbial mats to try to gain insights into what life may have been like back on that early Earth. And this is just a picture of some of the hypersaline mats that I studied as a postdoc down in Guerrero Negro, about halfway down the Baja Peninsula, in the salterns that are there. So the Mexican government and the Mitsubishi Corporation have a joint venture where they take seawater and evaporate it and generate those ponds that you may have seen, similar to the ponds that are near the San Francisco airport as you fly in the Cargill salt flats there. They often have a deep red color because of the microorganisms that bloom and really thrive in those very saline ponds, and this is an example of a biofilm or a microbial mat. And I just draw your attention to this. When you first pull it out, it looks like this, and it's very sulfitic and stinky and anaerobic down on the bottom. But on the surface, there are cyanobacteria or diatoms, so there's algae that are producing oxygen at the surface, whereas down here, it's stinky and anoxic. But if you let that mat sit out for overnight and come back and look at it the next day, you can see all of these layers, which I hope you will agree seem reminiscent of those stromatolite pictures that I just showed you a few minutes ago. And so these are modern analogs of what was going on there. And we study these modern mats and learn about the amazing diversity of not just the species of bacteria that live there, but also the metabolic activities that they can do. So microorganisms have always been fascinating to me because they can do it all. right? So we as humans, right, we may be a vegetarian or we may be a carnivore, but basically we need to eat our carbon, right, to survive and consume our nutrients and essential things through our diet. Well, many, many microorganisms out there do that as well, and they would be so-called heterotrophic bacteria, something like that. Something like E. coli that lives in our gut, right? But many microorganisms out in the oceans or all over the planet, they are able to fix their own carbon, or they are able to cycle things like nitrogen and sulfur in ways that we would never be able to do. And so tonight I want to give you a sense of some of that. And so a microbial mat, as I mentioned, is full of these diverse communities. And so as you go down from the surface down into the mat, you can see here the oxygen levels decline, the sulfur that I showed you, that black color, increases as you go down. But there's many different groups of organisms. Some of them are found at the surface and some are found as you go further down. And this seems to be a hallmark of these microbial mat communities in these consortia. And so there's often, and some of my own work in this hypersaline system showed that they can even migrate up and down from day to night as they modulate their light levels and their sulfur levels and all these sorts of things. So they're very adapted to these environments. So prior to taking my position at Cal State Long Beach, these were the hot springs, and these hypersaline springs were many of the things that I studied. I didn't focus on them as much, but there were chemolytic sulfur bacteria in those mats, and I just draw that to your attention because it's going to be relevant to the story here from our local sulfur mats here. So these are chemolithoautotrophs. And so what that literally means is they're rock eaters. So a green plant or a photosynthetic cyanobacteria or an algae growing like kelp off the coast here would be photoautotrophs, right? So they use the power of the sun to generate their energy and fix CO2 producing oxygen, which we of course really appreciate. But these organisms don't use photosynthetic energy. They get their energy from the rocks and minerals that are found in various places on the planet. And one of the more interesting locations for that is at hydrothermal systems, hydrothermal vents. Now it turns out that these chemolithoautotrophs, especially the sulfur-eating ones, were discovered centuries ago by a very famous Russian microbiologist, Sergei Vinogradsky, who lived from the middle part of the 19th century into the 20th century. And he actually was one of the first microbiologists that you might call a microbial ecologist, which is what I call myself. But we didn't have those terms back then, so he would call himself a bacteriologist. But he discovered this process whereby you could take CO2 from the atmosphere and create biomass or create life out of this, right? But instead of using plants, photosynthesis like plants do, these organisms are using nitrogen or sulfur compounds. And when you're talking about deep sea vents, you're really talking about sulfur. And I hope everybody in this audience, I'm sure you're all such big fans of marine biology and you've seen pictures like this or watched videos on the Discovery Channel or something like this, right? So the so-called black smoker vents, which are amazing, and you have to get in a submersible or an ROV to go down there, and I've never had the pleasure of doing that. And what you can see here, the red color and the white color, those are the tube worms that were discovered when we started taking submersibles like the Alvin submersible from Woods Hole Oceanographic Institute down to some of these vent systems, and we discovered that there were animals living down there, which was baffling to the science at that point, right? So how the heck can a worm live down there, in fact, thrive and form these massive colonies? Well, the clue is that they lived right next to these sulfur vents. And what we now appreciate is that these worms are special worms, and they really don't even have a normal stomach. Instead, they have a bag full of bacterial symbionts that are able to metabolize the sulfur being spewed out. You can see the dark color here, just like I showed you in that microbial mat earlier. So these worms have these symbionts that can feed them and fix carbon and allow them to live very happily down near these vents. Even though most tube worms that you would find off the coast right here have to be things like filter feeders, bringing in their food from the ocean and digesting it in their stomach. So when we discovered these systems, we realized that there was a whole other world down there. But we also appreciated that chemosynthesis or chemolithotrophy can be the basis of an entire, almost like oasis ecosystem on the sea floor. And go down with that. And so one of my questions was, are there ways to study these vents a little closer to home? And so that's going to be kind of the focus of our talk this evening. There's other environments where you can see sulfur chemistry in action in biofilms. So off the coast of Santa Barbara and sort of up north a little bit, you can see these cold seeps where things like methane are released from the earth, and these form the basis of an entirely different ecosystem, which is analogous to what we see with the hydrothermal vents, but very different in terms of what's driving it there. But again, we see these white organisms, and these white organisms are chemosynthetic or chemolithoautotrophic bacteria. So there's a number of places we can go on Earth to find these, and they're often in these extreme habitats. And when I first took my position at Cal State Long Beach, and I want to acknowledge Alan Miller, who is in the audience, we were just reminiscing. He was the chair of our department when I was first hired long ago. And when I first came here, I was a young faculty member, as you can see here. I still had a nice full head of dark hair, which, alas, time marches on. But I was enthusiastic to find new research projects and involve my students. And I got a call from Ed Mastro, who those of you who are friends of the Cabrillo Aquarium will know his long tenure here. So he reached out to me and introduced himself and said, you know what, we have these white mats that form in the salt marsh over by our aquarium over here in San Pedro. And I said, well, I've never been there before, so why don't I get in my car and drive over? And so I came over with some of my students and we checked it out. So we went over and we looked and we could see, so this is a picture taken from that very first trip, my first visit to Cabrillo Marine Aquarium. And if you scrape the surface a little bit, you'll see that dark color, which is that sulfur that I've been showing you in these other pictures. So it's very reminiscent of what you see in these hydrothermal vent systems. So me and my students kind of took some samples, and honestly, we played around with them a little bit, and we put them in the freezer, and we didn't pay too much mind to them for a while. And I got involved in other projects. But then I decided to do a sabbatical at Caltech. And when I chose the collaborator that I worked with up there, Victoria Orphan, we were talking about possible projects. And we got to talking about the fact that there are other places where these sulfur vents can be found. She had collaborated with me on some of those Baja hypersaline mats that are fueled by photosynthesis, but we got to talking about the sulfur vents, and I said, hey, you know, I have some ideas of where we might go study those. And so we really just went right around the corner over here to White Point. And we decided to study those because I'd been directed by folks saying, you know what, there's some here at the aquarium, but actually the ones here in the salt marsh kind of disappeared a little bit. They were a lot harder to find. But I was told, hey, over in White Point, these things are everywhere. And if you go at low tide, you can really smell the sulfur. So why don't you go check those out? And so I had a geology graduate student who approached me about doing a master's degree. And I said, well, you know, I think you might be interested in doing something more along the lines of what we would call biogeochemistry, right? So looking at the biology, but also how it interacts with geology. And White Point seemed to be a really great setting to do that. So Priscilla pursued a master's degree in my lab for a few years and graduated in 2015. So this project has been going on since the early 2010s. And then I had another graduate student that came along, and she decided, Jessica was a marine biology student who decided to stay on and pursue a graduate master's degree in microbiology. She wanted to switch fields a little bit. And so she approached me, and she had come with me for my marine microbiology field trip to White Point, and she said, you know, I really liked that. I was interested in maybe developing a master's thesis there. So I said, sure, well, you can build on the work that Priscilla did. And so tonight's lecture is largely going to be based on their master's thesis work. But before we get to that, I want to give a little more context here. So White Point is named essentially because it's got these amazing communities over there. And you can see them on the beach in the intertidal. You can, if you put a scuba on or a snorkel, you can see them in the subtidal. And the hydrothermal history in this area is pretty profound, right? So if you go to White Point and you look up the cliff there, you see the amazing geological stratigraphy or layering and all the warping that has happened as our coast has uplifted, right? So the geologists study all of that amazing stuff. But we have these layers that have been established, and they're very tectonically active. And so it creates an environment where you have hydrothermal fluids that are flowing up through the rocks. And back in the day, in the early times, in the early 20th century, late 19th century, there were actually, there was a Japanese onsen hot spring kind of resort that was found there, which has since left. Now when you go over there, you see the lifeguard station and the beach there, and then you can walk over to the rocky intertidal area, which is shown here. Now the reason that there's sulfur here is because of this geological input. It's a little different than what you see in the deep sea. So the sulfur that we get here is actually a result of the Monterey Shale formation. And those of you that are aware of the oil industry around here will know that the oil we pump around here is Monterey Shale oil. But that oil is filled with sulfur, right? So different kinds of oil have different chemical compositions, and it's not just the oil particles in there. So as hot water is heated up and flows up through these geological layers, it picks up the hydrogen sulfide that's in there and brings it up. And then it's released at the surface and forms the basis of an analogous system to what we saw in those deep sea vents. So I've been bringing students out for field trips here for quite a few years at this point because it's such a fun thing to do in my marine microbiology class. And I very quickly noticed that there were these, if you turn over a rock like you see here, you'll often see white on there. And it looks very filamentous, and that's for a reason. So the bacteria that grow there, and I'll show you lots of pretty pictures of these, actually form long chains or filaments, and you can even see them with the naked eye here. So this biofilm is a little different than some of the ones that I showed you earlier. It's very light and feathery and easily disrupted, but it's visible with the naked eye nonetheless, which as microbiologists, we're often really happy to not have to look under the microscope at everything, so we appreciate that. So this is an intertidal area, and when the tide is low, you can go in there and really smell the sulfur, and you can see these rocks and uncover them. And then of course when the tide comes back in, they get covered up. And then if you dive down a little bit offshore there, just inside where the kelp beds start there, you'll see these subtidal vents that are always covered by water, not the same as the intertidal ones, but the similar organisms live there. Now here you can see this yellowish golden color. Those are diatoms that are living in a biofilm on top of these rocks. But then where the sulfur is coming out in these cracks and little areas between the rocks, this is where you see these biofilms forming. So we were really interested in this as well. All right, so let's talk about the microorganisms a little bit. So we as microbiologists, it's a little hard to characterize bacteria in terms of their species names. It's very challenging. So we tend to try to group them based on what they do. And so there are many different organisms that are capable of doing this, and I'll show you some trees of life to kind of give you a flavor for that a little bit later. But there are different types of bacteria that are capable of doing what's called sulfur oxidation. And these are those chemoautotrophic organisms that I was talking about earlier. So the hydrogen sulfide bubbles up from the earth, whether it's in a deep sea hydrothermal vent or here, and it comes out into the oxygen. And so these bacteria are really fascinating because they're able to take the hydrogen sulfide, which is highly toxic to aerobic organisms like us and many others, and they grab oxygen from the seawater and they bring them together and perform chemistry. And so basically what they're doing is they're converting hydrogen sulfide into sulfate. And that generates energy. And they can then use that energy for a lot of purposes, but one of those things is to fix atmospheric CO2 and turn it into cells and biomass. So they don't use the light's energy like a photosynthetic organism does. They use this reaction to generate the energy they need for this. And so this is just a picture under a microscope of one species called Beggiatoa that form these long filaments, and these are these sulfur oxidizers. And so many of these will have names like thio, which refers to the sulfur group, and things like thrix, which means thread, right? So thiothrix is a thread full of sulfur, or the thread that uses sulfur. And when you look under the microscope, you can actually see inside these filaments, you see all these little white globules. So those are sulfur. So what I didn't show on this equation, because none of us want to be too bogged down in the chemistry, is that they convert the H2S to elemental sulfur, and then they convert it to sulfate. And inside their cells, this is the elemental sulfur, which actually gives the biofilms that milky white color, because elemental sulfur looks white. If you look at it under the microscope like this, it almost looks pearlescent, but to the naked eye, it looks white. So one thing about microorganisms is that if they can do a pathway in one direction, they have almost undoubtedly evolved the ability to do it in the opposite direction. It may not be the same group, and often it isn't the same group of bacteria or microorganisms, but they often synergize by living together. And so one of our questions here, as I'll come to in a minute, is are there sulfur or sulfate reducing bacteria that live with the sulfur oxidizing bacteria? So basically, can you go one direction and back the other direction at the same time in these systems? And that's been demonstrated in a number of other systems. So one of our questions was, is that going on here at White Point? And this brings me to Priscilla's research questions. So with microorganisms, you can't go out and look at it with the naked eye and say, oh, that is this kind of animal or plant or kelp or whatever you're looking at. So you can look at it on the microscope, but even doing that can not always give you an identification. So we tend to rely, as I'll talk about today, on things like gene sequencing and other more modern technologies to try to identify these bacteria. And what that means is that if you go to almost any environment, whether it's the ocean or on land, and take a sample, you don't know what's in there. So you really have to ask the question first, who's there, before you can really ask, what are they doing? And so as microbial ecologists, that's often what we do. Now in this case, we have a strong indicator that there's sulfur cycle going on, and so that's what we wanted to probe with her. So what other groups are living? But undoubtedly, we predicted there would be non-sulfur players in this consortium as well, based on what we had seen in those hypersaline and hot spring vents and things like that. So our question was, are those guys that go backwards, the sulfate reducer is abundant, and are they active? And then how do they relate to each other if they're both found there? And so do they form a cycle, in other words? Does one organism produce through its metabolism a product which then becomes the reagent or the reactant that drives the opposite metabolic reaction? And so those were kind of her basic questions. And more generally, she wanted to ask the question, how do these white point vents compare to the deep sea vents? And so she did a lot of work comparing the data she was able to get from here with what other researchers have gotten from the deep sea systems. Now, looking at these systems over here actually has an even longer date than when I first came here. And Dr. Miller will remember that there was a former graduate student in our department, Jeff Stein, who actually was curious about these mats long before we were. And he published his master's thesis looking at, now we don't have a picture with the abalone eating them like he was studying, because as many of you are aware, the abalone are in rare supply these days. But now it appears that sea urchins move in and may be grazing on these biofilms as well. So these seem to form maybe part of a broader ecosystem, so I always like to bring that out. We have not engaged in research looking at the animal interactions in our lab, but, and incidentally, you can even see them growing on animals that live down here in these cracks and crevices where the vents are. So the bacteria are happy as clams or happy as crabs in this case to just hang out on the back of there. So I mentioned some approaches, but, and I'm not going to go too far into the weeds on these, but I want to give you an overview of some of the methods that Priscilla used. So she used gene sequences, as I mentioned, to try to get at the diversity of the organisms living there. She also used microscopy to try to take an image and look at that. And so there's a method where you can combine the gene sequences with microscopy and come up with these amazing gene probes where we can light up certain cells of a specific species while we're not lighting up others. So I'm going to show you some images that give you a sense of, oh, we can probe or ask the question, what is that cell, and is it different from the cell that's next to it under the microscope? And so what she did is she actually put out some slides into the intertidal area and watched what grew on it, and you can see the white filaments that grew here, and there's also some, looks like some algae and phototrophs that grew there as well. And we wanted to know if they were active, and so it turns out that you can use radioactive sulfur as a tracer to determine if sulfur cycling is going on. And so she was able to do that as well. So I don't want you to focus too much, and I know it's a little bit blurry on the screen here. You don't need to see the species names or whatever, but I just wanted to give you a sense. When she started doing gene sequencing, her first question was, how many of these sequences that we get back are identifiable as a sulfur bacterium, right? And what she found was a lot of them, right? So it was pretty amazing, not surprising, but there's a broad diversity. But the technique she was using here was fairly limited in terms of the number of sequences she could get. She did it again, and she was able to identify that some of these organisms with the red arrows are those sulfur-reducing bacteria, the ones going back the other direction. So she identified both the oxidative and reductive pathways. So we have a full cycle going all the way around that little wheel I was showing earlier here, which was pretty cool. Now, in the time since this project first started and more recently, we've had an amazing revolution in genomic sequencing technology. And this is what allows you to send off your 23andMe and get your answer back on, in like a week or whatever they do, right? So next generation sequencing has revolutionized genomics as a field and allows us to get a lot more data from these systems. And so this was coming about just about when Priscilla was doing her master's thesis. So she did a little bit of this at the end, and then I'm going to show you more results from the later students. So she did 2 samples of this, and instead of getting 100 sequences, she got 16,000 sequences, so that was pretty cool. And she was then able to go in, and by more fully sampling them, she was able to really get out the diversity. So this is like if you went to the Amazon, and instead of just identifying the major tree canopy species, you were able to get down in the dirt and find all those little critters that might be hiding, right? So we can now do this with the microorganisms as well. And so what she found was there were similar diversity in groups, but for example, the one on the left here in the white is Marathrix, which is one of those sulfur bacteria, and it was about 1/3 of her library of sequences on the left-hand one, and then on the right, it was much fewer, much smaller. So there's some variation here, which is not a big surprise. We see this when we take unique samples and analyze them, and in this one, you see this light blue guy, sulfurovum, was more abundant. Well, that's another sulfur bacterium, but a different type. So there seems to be a lot going on here in these very diverse systems. And we're going to explore that some more later on with the work of the other students. So she, at this point, had identified that, yes, there's a lot of diverse organisms here. There's both the sulfur oxidizers and the reducers. But we really wanted to see it with the naked eye, because looking at gene sequences all day kind of makes your eyes go squinty, and it's not terribly exciting, honestly. So she did this in situ hybridization fluorescence microscopy to get a chance to look at that. And so what she did is she took the gene sequences she had obtained, as well as some others that were just routinely used in the field, and she created or purchased these fluorescent probes, which basically allow you to go in under the microscope and ask the question, is that cell this thing, or is it a different thing? And so she used a range of probes that would be targeting those sulfur bacteria to try to get at some of these questions. And so this is just one of a number of pictures I'll show you. And what you see is there's an amazing array of diversity, of the morphologies of the organisms. So here we're seeing these clusters of unicellular bacteria, these green ones. So these green ones are the gamma bacteria, the gamma proteobacteria, which is the group, one of the major groups that you see the sulfur oxidizers in. And then that reddish one there that the other arrow is pointing to here are delta proteobacteria, and those are the sulfur reducers that go back in the opposite direction. So now, not only can we see it on our computer from our gene sequences, but here we can look under the microscope and look at the associations and the relationships of these. So she did a series of studies looking at this. And so I'll just kind of walk you through this. This large blue thing here, this sort of glowing filament, is that Marathrix that I mentioned earlier. So that's one of those proteobacteria that we know is a sulfur oxidizer. And so it's living here in filaments, and there's big filaments, and you can see the little filaments, and those are likely different unique species, right, of bacteria in that group. And then the red guys over here are, again, those delta proteobacteria. So some of them seem to be doing their own thing, right? But of course, at the microscopic level, they're still interacting with one another. Now, one group that was particularly interesting to us was something called desulfuramusa, because this was one of the major groups of sulfur reducers, one of the ones that goes back the other direction. And so she developed a probe. This was her unique probe that she did. And when she probed it, she found these really cool, like, filamentous organisms. So the reddish filaments poking up kind of like, there's, this is so long that it's hard to focus on them because they actually span multiple layers of the focal plane when you look under the microscope, right? So if you've ever looked under a microscope and you go up and down, you can kind of see how the image changes. And so this is kind of an example of that, where it's almost like they're poking up out of the bottom of the slide there. So she was able to identify those organisms and find out what they look like for the first time. And she found that they were found right next to some of these clustering, clumping gamma proteobacteria. So again, there seems to be some of these physical associations. Now, if we zoom in on some of those big filaments like I showed you earlier, so the Marathrix, that big blue filament, here we're zoomed in, and this is one of those filaments. But what you can see there are some delta proteobacteria and the desulfuramusa. These are all the organisms that go back the other direction. They're living on the surface of the Marathrix. And so the obvious question there is, are Marathrix using the sulfur, producing sulfate from that, and then is it going right back the other direction by the desulfuramusa? Or in the case of the desulfuramusa, are they using the elemental sulfur? Remember I showed you those little pearly looking globules inside the cell. You see those in Marathrix as well. And it turns out that desulfuramusa, they can't use sulfate, but they can use elemental sulfur. And so what we think might be going on is the Marathrix produce the sulfur, release it into the environment, and then they metabolize it back the other direction. And they might be along the lines of what we know from other fields as an epiphyte, right? So if you study plants, you know that oftentimes there's microorganisms that live on the surface of the plants. Sometimes it's algae, sometimes it's bacteria and other things. So this might be a similar, analogous kind of situation where you have the big thing with little tiny things living all over the surface, mooching off of it or coexisting with it in this consortium that enables it to survive. And so really the last thing she wanted to do was to identify whether they were active. And what she showed was, using the radiotracer, she showed that they are active. And what she had to do was place strings in the intertidal right here, and she let them grow. They sat them out there. The biofilm grew on the strings just like it did on those glass slides. And then she brought them in, and they looked like this. And then she performed the assays asking the question, are they active or not, back at CSULB in the lab. And I won't go into all the details here, but basically what she showed was on 2 separate occasions she was able to demonstrate that there's active sulfur reduction going on in this system, meaning that that backwards reaction is indeed actually happening and is a biological reaction. The reason we know that is that our controls here, which are the things with no bars over on the right-hand side, didn't show a bar. And interestingly enough, if you compare this with the rates that I obtained from that hot spring in Yellowstone National Park I showed you earlier, these rates are very similar to rates that I got in that spring. So this seems to be an important process in this system, which was pretty cool. Both chemosynthetic and photosynthetic mats have this. So as just a quick summary of Priscilla's work, she found this very diverse community with lots of sulfur action going on, including these sulfur oxidizers and reducers, very high rates of sulfate reduction, which might be fueled by the carbon fixation and the chemosynthesis inherent in the system. And she concluded that these nearshore vents are actually pretty similar to the deep sea hydrothermal vents as well. Now Jessica came along and followed up, and her question really springboarded right off of that. And she asked the question, are intertidal mats similar to the ones you see in the subtidal? Priscilla didn't sample the subtidals at all, and so Jessica wanted to get her wetsuit on and go down and collect some samples, and she did that. And the other question was, do they vary over time? So she also sampled at White Point, and she sampled in March and June of 2015 in the intertidal and subtidal. And one of the reasons we had this question is that you can notice the subtidal mats are very fluffy and feathery looking, whereas in the intertidal we often get a film that forms, but it doesn't have those fluffy filaments. And that is probably because of wave action in the intertidal, right? So it washes away those filaments potentially, or the same things. And so, to make a long story short, what she found when she did that same gene sequencing that Priscilla did in the intertidal in March and June and the subtidal in March and June was that you saw different communities that formed, and that there were time and spatial differences, but that the subtidal communities ended up being more similar to each other than the intertidal ones, at least when looked at in the aggregate. And some of these major players are things we've already talked about. So this sort of light blue one is that sulfur ovum that we talked about. And some of these purple ones are some of the other sulfur bacteria. Now, if we look in aggregate, it's a little easier to see this. So if you just ask the question, are these similar in terms of their diversity, right? So we look at species diversity as an important metric in ecology. And so when you did this, when she went out to the intertidal, she found that there was lower diversity in one of the March intertidal one. And then the letters here indicate which ones are significantly similar to each other, right? So the March one was totally different than everybody else. The subtidal March one was totally different than the intertidal one and all the other ones as well. And so what you end up with is a picture that there's something different going on here in the subtidal and the intertidal, right? And so lastly, she did some analyses looking at sulfur and anions like chemistry in these vents to try to get a handle on that. I'm not going to really belabor that, but she found some differences. So to summarize what Jessica found, she found that the mat communities in the intertidal and subtidal had similar groups of bacteria, but they were different in the aggregate compositions. And there seemed to be greater variation in the intertidal over time, and you saw that March intertidal one was much lower than the other samples. And that environment probably plays a role. We didn't get as far down the road of determining all the chemistry of that as I might have liked, but it's very challenging to collect chemistry data from the vents in the subtidal where you have to like get down there and sample. Excuse me. All right, so the last little vignette I want to share with you is some more recent work, and this is work by one of my current master's students, Natalie Kokel. She's got her fingers crossed that she's going to have her master's degree in spring of 2024. So what she's been doing as a side project, her project is actually looking at the guts of some of our burrowing heart urchins off the coast here as well. But she's using these genomic approaches, and so I talked her into analyzing some of the data that Jessica had gotten years ago but never been able to analyze. And so what we're doing here is instead of just looking at one gene at a time, which is the data that I've showed you before, We're looking at all the genes. And so it's really a whole genome approach. And you can do this with a pure culture. So like if you had a strain of E. coli that was growing in your gut and you wanted to know if it was bad for your health, you could culture it and sequence that genome. And the way that works is you basically chop it up into little bits, you sequence it, and then you ask a computer, let's line these up and form what we typically show as a circular genome. Turns out that most bacteria, that's the shape of their genes. You know, we have our 23 pairs of chromosomes that, you know, have our sister chromatids and all that good stuff you learned about in high school biology. Bacteria usually have a single or sometimes multiple circular genomes. But if you're going to analyze a mixed community like these microbial mats, then you're really asking a question about multiple genomes all at once. And so you need a lot of computing power and a lot of what we now call bioinformatics analyses to be performed. And Natalie has been a real trooper in learning how to do this. I'm such an old fart that I don't even know how to do these analyses because they didn't exist when I was a graduate student. So fortunately, we have some newer faculty that are able to help her with those analyses, and she's been teaching me as much as I've been teaching her with some of these aspects of the project, which is really fantastic. And so when you line them up with a mixed genome, you have to make sure you're not lining up the yellow ones with the purple ones with the red ones. You want to separate them out somehow. And she's been able to do that. I'm not going to show you how that works, but she was able to come up with an overview for this talk of some of what she's found from that. On the left-hand side are some pathways, and basically what you can do is submit the sequences to databases. This database is actually housed in Kyoto in Japan, and it has millions of gene sequences or billions of gene sequences at this point, and you can take your genes, send them to that, and basically ask the question, what's in my genes? And it spits back a pathway map like this, and it does that based on how similar the gene sequences are that you have with something that's already been characterized in the database. And so what she's able to do is ask the question, this pathway map has been worked out with other organisms that we're not studying, like pure cultures of organisms, but we can ask, is that found in our environmental sample as well? And what you can see here for carbon is that all of the boxes are red, and what that means is that it has every aspect of each of those pathways. Not surprising for this, because these are carbon fixation, and I already told you that these are chemoautotrophic mats. So they are fixing CO2 and turning it into biomass, right? So we would have been shocked if there weren't all of these genes here. And in fact, there's other alternative pathways that are also found. So there seems to be more than one way to fix carbon in these mats, which is pretty exciting and interesting to look at. But sulfur is really the story that we're most interested in here, and so this is what we're looking at. And what she's been able to find here is a little bit more surprising than the carbon story. So she's been able to find that the sulfate reduction and some of the sulfur oxidation pathways are there, and that's found here. But it turns out that one of the pathways, the SOX pathway, was absent, or at very low levels at least, it wasn't coming up as being red on this pathway map. And that was a big surprise to us, because based on that tree that we saw with those groups that were being identified as sulfur oxidizing, we thought we had these genes in here because of who they were identified as. But what this shows is that you really have to get at the genes themselves, the whole genome, to understand what's going on in a system. And so they may not be completely absent, but they're much less common than we expected them to be, which was a bit of a surprise. And so to summarize Natalie's work, what she found is that intertidal and subtidal mats have very similar compositions. I didn't show you it, but basically when she compared, she did the same thing Jessica did. Jessica found some differences between intertidal and subtidal, but it turns out with the whole metagenome, we didn't see much. And that's probably a function of the fact that the diversity of the metabolic pathways is so great that they all just kind of blend together. So we were somewhat surprised by that sulfur pathway that I mentioned, but not so much by the carbon. We also found pathways of methane cycling and nitrogen and photosynthesis as well, so it's unsurprisingly very diverse, and we'll be exploring that some more. All right, so I think I've already gone over time here, so I'm going to try and wrap this up and give you a few take-home points. So the work that these amazing students have done out here at White Point has shown that we have an amazing array of diversity of microorganisms. The rainforest in the teapot was really found in this set of mats as well, and I wasn't surprised, but it's always amazing to get in there and discover exactly what's there. The sulfur bacteria are really important, and it seems like we have this, both the oxidation and the reduction pathway. So we have a complete sulfur cycle here. And that was really the question we were asking with the project. I hope you agree that we've demonstrated that we can study these mats, these vents, and they really are quite similar in many ways to those deep sea vents. So we can just drive right over to White Point, and we don't have to book time on a submersible. Now, since I started this work, and my students have started this work, I get emails periodically from folks that take video, and they found out about this crazy professor at Long Beach State who's been looking at the vents, and so they'll send me videos or pictures, and they'll ask, are these the sulfur vents? And they've been found all the way down to San Diego and all the way up north to Santa Barbara. So these seem to be very prevalent, and it would be wonderful to have a student that could actually go and sample all up and down the coast. And we could do something along the lines of what is the geography of the organisms that are living there, but we haven't had a chance to do that yet. And so lastly, I'd just like to once again acknowledge Priscilla, Jessica, and Natalie for their hard work here, as well as the many amazing undergrads that I've had that have worked on this project, and many others in the lab, of course. You know, Cal State Long Beach, our mission is to educate undergraduates and master's students, and our marine biology program is very proud of how many students that we're able to invite into our research programs and send them off into careers in marine biology, and including right here at the Cabrillo Aquarium. So we're just so wonderful to have the relationship we do with our alumni over here. I want to acknowledge the collaborators. Victoria Orphan, who I mentioned at Caltech, was really a great collaborator, as well as her postdoc, Roland Hotzenpickler, who's now got a faculty position at Montana State. We were funded by the National Science Foundation, the Geobiology and Geochemistry program, as well as the CSU Coast organization for this work. And with that, I will take questions. Thank you very much.
Thank you, Dr. Dillon. Are there any questions for Dr. Dillon?
All right. Well, in the back there, we'll...
The hydrothermal vents, are they a result of volcanic or something activity? How far from shore?
Yeah, so the hydrothermal vent, the question was, are they the result of volcanic or sort of, they are the result of the tectonic activity, the seismic activity having on the coast here, right? So it's not a volcano per se, and it's not the same as what you would see in Yellowstone National Park, where you have an amazing array of geothermal features. But because of the tectonic uplift here, it's created these fissures and cracks, and so the high pressure, high temperature fluids from down deep in the earth are able to escape. And so as they percolate up through, they're percolating through those oil-rich layers, picking up the hydrogen sulfide along the way, and then as they get released into the ocean or the air, if you smell them under the low tide, then you smell that sulfide being released. So, and I'm not a geologist. I know there's quite a lot of geological work that's gone on here in terms of how deep they might be coming from, but those fluids are probably moving up quite a long distance. Now, I should note, I forgot to mention that the fluids coming out here are not nearly as hot as what you see in some of those deep sea hydrothermal vents. They can be as high as 300 degrees Celsius. Now, as you'll hopefully remember, we can't have 300 degrees up here at atmospheric pressure, but on the deep sea floor you can because of the amazing pressure that's exerted by the water on top of that. So at our temperature, 100 degrees is boiling and it would just boil off, right? These aren't even close to boiling though. They're actually more what you would call them sort of a warm spring, but it's really the chemistry that's being released that seems to be driving these chemosynthetic systems. But interestingly enough, they look very similar when we compare the communities here with what you see at 200 or 300 degrees Celsius on these deep sea black smokers. So the chemistry seems to be driving it more than that. I know I went a little beyond your question there, but yeah.
Essentially two questions. One is that I'm a little confused. The first colonizers of land, you're saying were the mouse and not the fungi?
So we think that there were...
Remember, Dr. Dillon?
Oh, yeah, sorry. Yeah, thank you. So the question was, who colonized land first? Was it these microbes that I've been talking about, the bacteria, or was it fungi or something like that? The biofilms that I'm talking about that date back billions of years were likely in shallow water habitats, and so you wouldn't really call them a land colonizer. Now, when life really came onto Earth, came onto land as we think of it today, right? So you had the Cambrian explosion with animals developing, and then later on life came out of the oceans onto land. The microbes were already on land at that point, right? So we don't know, I guess the not satisfying answer is we don't know which microbe colonized land first. We do know that fungi came after these bacteria in the evolutionary history. Eukaryotes came, billions of years later, but still have been around, at least 500 million years on our planet. And so, yeah, that's an uncertain question. And of course, the microorganisms that colonized land really then helped pave the way for the animals and the plants to come later on. You had a second question, or?
One is, do a lot of the bacteria have the pathways present to either oxidize or reduce sulfur, and you just need the right environment in order to
Yeah, I see. That's a good question. So he's asking, are these sulfur bacteria, or the genes at least, present in a lot of different locations, a lot of places? So there are definitely sulfur bacteria found in many different habitats, and one of the sort of basic principles of microbial ecology, for a long time there was a famous microbiologist, Boss Becking, who said, everything is everywhere, the environment selects. And what they're getting at there is that the diversity of microbes and the way they can spread around the globe is so prodigious that they really are everywhere, but you don't see them in large numbers everywhere because in a given habitat, the conditions are not right for them. And so that essentially is the answer to your question, is that there are these kinds of sulfur organisms probably all over the ocean, but they're not real happy in cold water. And one of the basic questions we have, whether it's in a hydrothermal vent or in a hot spring, is you see this amazing array of organisms, and if you go from this spring to that spring to that spring, you see the same organisms there. But how do they get from that spring to the other spring? And so there's a number of theories about this. One is that they may go through the plumbing, down through the earth. But of course, if the temperatures are exceeding 300 or 400 degrees Celsius down there, it probably exceeds the possibility of life. So that gets a little dicey. So maybe they disperse through the ocean just like animal larvae do, right? So they float around, and maybe they can form spores or other structures, or just hang out and go dormant while they transit from one location to the other. And I know a number of years ago there was a talk by Shana Gaffredi from Occidental. She studies whale falls, which actually have a lot of similarities with the hydrothermal vent systems. And there's sulfur bacteria that live on dead whales on the ocean floor, and so there's a thought that there might be like a stepping stone effect where vent organisms can live on whales, can then find another vent somehow and make their way around. But it's a bit of a mystery how they're dispersed, that's for sure.
That's so weird.
Yeah.
Do microbes come from space?
Can microbes come from space? So there's an entire field of astrobiology that is very readily addressing that. I was actually funded through NASA and an astrobiology fellowship for my postdoctoral work. I tend to focus on the life we know that lives here, but there's a lot of really bright scientists that are out there trying to figure out. And of course, as I hope you're all aware, NASA is sending missions to Mars, and the long-term plan is to try to recover samples there, bring them back to Earth, or go there and detect if there's life that can be sampled there. We know that the Earth is covered with space rock, right? And that space rock has carbon in it. So every little bit of meteorite or whatever that encounters, burns up in our atmosphere, delivers a little tiny amount of carbon into our Earth. And this may have played a role in the generation of the carbon necessary to get life going on our planet. And there is a theory that life, when it arose here 3.8 billion years ago, which is our nearest estimate, actually didn't evolve here. It came from somewhere else, right? And we don't know. So that would be a panspermia theory, where maybe life originated elsewhere in our solar system, or certainly you could extend that even further, and arrived here on Earth at a time when the Earth's environment was suitable for life to grow. So that's another possibility that is being explored. Do we know that that's happened? No, but it's possible. And as scientists, we keep our minds open to the possibility that, because really we don't know exactly how life evolved here to begin with as an alternative, right? So there's theories that have been postulated for centuries now about how life may have evolved. A lot of it does center around hydrothermal vent systems as a possible cradle of life, or what we now know as the deep subsurface under the crust of the Earth. These environments are very attractive for a number of reasons. One is that they're protected from that ultraviolet radiation that I mentioned earlier was so strong on the early Earth. But if you're on the bottom of the ocean, the UV can't penetrate through the water. Or if you're in rock fissures inside the Earth, it's not going to make it there, right? So those, and the chemistry that can occur in these deep subsurface systems is very energetically favorable, which is why you see these amazingly diverse communities here. So if we're pondering how this might have happened, you've got kind of the building blocks of life, but we don't have the missing link in terms of exactly what that early life looked like. And so we're still exploring that. We frankly may never find that perfect answer for this, but we continue to research it to try to understand. And there's an interesting other thing. When you do the tree of life with these gene sequences, it turns out that thermophilic organisms tend to be found at the bottom of the tree of life, near the root or the origin of life. Now, there's some reasons why that may be an artifact, but it's also possible that this reflects the fact that the earliest life was thermophilic, and so that's why you see it near the root of the tree of life, which then went on to diversify and split off into the billions of groups of microorganisms that likely inhabit our Earth today. Sure, down front.
I apologize, this is a sort of a lowbrow question. I have an extensive diving history off Palos Verdes Peninsula as part of my work. Only once have I encountered a hydrothermal vent, it was off White's Point, which is where a lot of the diving was. This is back in the 70s, and I remember three very impressive things: seeing the hot water coming out of the bottom, seeing that virtually all the hard substrate and invertebrate animals and the algae had sulfophilic bacteria growing on it. But the most dramatic thing for me was that a cabezon, demersal fish, had a cover of sulfophilic bacteria that I wish my hair today on my scalp was as thick.
Wow.
On that fish. When you see, or your students see, hydrothermal events, do they see the bacteria growing on fish? Yeah.
You are the first person to tell me that you've seen these biofilms growing on a fish. That's pretty amazing, just because.
That was the only fish.
Yeah.
Lots of other fish there.
Yeah.
But
And it was a living fish swimming around?
living fish.
Wow, that's pretty amazing, because you wouldn't expect that, typically you'd expect it to be something that is relatively stationary or sessile,
Yes.
and would sit there long enough for these things to grow on it. But I suppose if it was maybe that's evidence of feeding behaviors, and if this fish has learned to hang out near those vents for extended periods of time, the scales of a fish are a substrate that a biofilm can grow on. And fish have biofilms on them. Every fish in the ocean has a biofilm growing on it. It's just not usually something that you can identify as one of these sulfur. So that's really, thank you for sharing that, because I had not encountered that before. I showed that picture of the crab and obviously some of these other invertebrates that don't, they tend to sit there. But that's really cool. It would be wonderful to try and check out what's going on, but if you ever see it again, let me know. Yeah. I think there was maybe a question in the back.
I have a handful, but I'll only ask like one. We can chat later.
Sounds great.
So you were talking about the desulfuramusa and how that is almost directly intertwined with the marthrids. And I was wondering, so is it kind of like a chicken and egg thing where like one has to exist in an area for the other, or is one more dependent on the other?
Yeah. So the question is really like, what is the relationship between the Marathrix and the desulfuramusa, and how are they dependent on each other? And your chicken and an egg analogy is apt. If I had to say, I would think that the sulfur oxidizers, because that's really the main driver of the chemistry in the system and the metabolism of the system, probably preceded and would create the conditions under which they make that elemental sulfur, which then the desulfuramusa can use as their food source and go back the other direction. But of course, I don't know, which came first. I just know there's a lot more of the sulfur oxidizers that are present there. So that would be my best guess.
Because it's harder for them, the desulfuramusa, to just get the sulfur from the vents itself, because the elemental sulfur isn't as readily available?
Yeah, so the vents are releasing hydrogen sulfide, and some of the sulfur bacteria can use the sulfide, right, and oxidize that to sulfate. And seawater has plenty of sulfate. So there's the bacteria that can use sulfate and convert that back to sulfide, and they're found all over the ocean. So if you dig into the mud on the bottom of the seafloor, sulfate reducers are living there. And that's why you can get that sort of rotten egg smell if you wander out into a mud flat and put your boot in too far and you get that smell coming out. That's sulfate reduction. That happens everywhere. But the sulfur reduction is maybe more niche in terms of being found in these more specialized areas. As I showed you, these organisms collect and produce large amounts of the elemental sulfur, and so that may favor that. Incidentally, they do that because it's kind of like a bank account. It turns out that if they run out of sulfide, the sulfur bacteria that are the oxidizers can then eat up the sulfur globules inside their cells and convert them all the way to sulfate. And so if the sulfur vent disappeared for some reason, right? And that does happen, like I mentioned over here, the vents kind of come and go, and I haven't been out there in a long time, but they may not be as robust anymore. So if you turn off the sulfide coming out from the geology side of things, then they would be forced probably to eat the sulfur and move that along in the pathway. So the cool thing about these pathways is they involve very complex metabolism with lots of different enzymes. And in microorganisms, they often live side by side. So one organism makes the product, and then it's like an assembly line. So maybe something makes the sulfur, and then somebody else takes the sulfur a little bit further and a little bit further. So that's one of the things I've always been very fascinated with microorganisms.
There was one, but you kind of answered
All right.
it. Yeah.
Great.
Anything else from the audience? Yeah.
Okay. What? Yeah, sure.
Let's put answered that.
Any like follow-up studies, or is it known what causes the variation in the microbacterial communities composition?
Well, pretty much what we've done is what I showed you tonight. So I was kind of trying to show the thread of how we've followed things up. So we haven't been able to go back and readdress every question, but really Jessica's work kind of built on the genetic work in terms of the sequence diversity more than she followed up. She didn't do any of the microscopy or the sulfur rate stuff. We haven't really followed that up at this point, but yeah, so there's definitely further work that would be, there's always other questions and that's the wonderful thing about science, right, is, and so we'd love to follow that up. So maybe I'll have a future student that works on that someday.
How long have you been studying this?
Well, so Priscilla's work ended in 2015. So her thesis probably started in, I want to say like 2011, something like that. But it was probably 05 maybe when I came over and visited with Ed and first saw the mats that were found over here. It kind of just languished on the back burner while I was exploring some other projects with my students in the meantime. So it went about a decade and then I got that going. So maybe it'll go down for a decade and then it'll come back again towards the end of my career or something. But yeah.
Are you the first to be studying this, or?
Well, no, and I didn't really go in. There have been some other bacteriologists that have studied. There was a lab that came and studied the bacteria from White Point area. So that was a lab from UC Davis, and they cultured bacteria, these Marathrix type organisms from White Point and studied those in culture. So I'm not the only one who has studied this, but and certainly there's many studies of similar systems throughout the world, but I think we've probably spent the most time mucking around out there in terms of the bacterial mats, at least at White Point.
Do you collaborate with other countries in this?
I have not to this point, but I would certainly be interested and open to that. There are some other studies, and when Priscilla was reviewing the literature to compare her system with others, she looked at the deep sea systems, but she also compared to other nearshore systems. And it seemed like our system here is quite a bit different than those that might be found in, say, the Mediterranean that have been studied, or in Asia or some other areas. You know, all around the Ring of Fire, of course, we have these tectonic activities, and so there's likely, you know, vent systems in all of the countries around the Pacific, and so it's much more widespread probably than we know at this point, but yeah.
I actually have a question. You touched on this, but can you elaborate a little bit more? Why do you think the sulfur flats in our salt marsh come and go, where the ones at White's Point seem to be more stable?
Yeah, and so I don't know, but these are soft sediments over here, and so I don't know what the underlying geology looks like there. But what I do know from my time working in hot springs in Yellowstone National Park is that you could be studying a spring. Now, some of them, like the one I showed you the picture of, Mushroom Spring, is very large, and so it's been around a long time. But there are actually smaller systems, smaller hot springs, that my PhD mentor, who'd been studying Yellowstone hot springs for 40 years practically by the time I was in his lab, he would take us around and show us, oh, you know, that spring used to be active and now it hasn't been for the last 5 years. Whereas this spring is new over here. It just sprang up maybe 5 years ago. So the plumbing changes over time, right? We've got these fluids coming up, and they move to find kind of the easiest path, right? And so that's probably involved in this, and it may be that there's something that's blocking the fluids from coming out over here, or maybe the source was shut off somehow. Because we're talking about fluids that are coming up from very deep, right? So, you know, hundreds of meters down into the earth, maybe even kilometers, when it first starts, and it sort of percolates its way up, and then it's kind of moving through cracks and making its way. And with the rocky intertidal area being an active uplift zone, then it's probably more viable that these cracks and crevices would be long-lasting.
No changes.
Whereas over here, maybe that it's not as active in the marsh, and that's going to be evidenced by the fact that you've got a mudflat that grows there, right? So clearly it's not lifting up in the same way it is right around the peninsula over here. So, but if you go over to Cabrillo Beach, we were also able to see some things right on the far side in the intertidal area there as well, way back when, but when I came back, those also had kind of disappeared. So it's a changing and evolving thing over time, and that's one of the fascinating things about it.
Any other questions?
Yeah, in the back.
What would happen if, you know, you poured chlorine, like the sulfur?
Yeah, so what would happen if you poured chlorine or maybe some sort of chemicals into that, kind of like you would in a pool, right, to kill your algae or whatever. So, you know, it get diluted really quickly. If you did it at low tide in some of those pools that I was showing you, it might kill everything, right? Temporarily. But then as soon as the tide came up, it would wash out and be diluted and it'd go away. But that does bring up, so there were some studies done in the early days. This would never be allowed today by the National Park Service. But back in the day of early Yellowstone microbiology, there were professors that will not be named that would do experiments not all that different than what you're suggesting. Maybe dump some chemicals in and see what happens to a hot spring. And partly they were interested in knowing like how long the residence time of of the fluids in those would be. So you're definitely not going to kill the microbes permanently there, but you might, take out just like you have to keep adding chlorine to your pool, you're definitely things will grow back for sure, but obviously that could have a local impact, but